Hello everyone, and a warm welcome back to our Evolution and Ecology seminar series. My name is Elizabeth Duxbury, and I'm a postdoc in the research group of Alexei Maklikov at the University of East Anglia. I'll be your host for today's session. Myself and our wonderful team of co-organisers, Andreas, Ulia and Wouter, in the groups of Judith Mank and Simone Imler at the Universities of British Columbia and of East Anglia, are very grateful for the enthusiasm from the research community for this initiative, and it has been a pleasure to hear from such a diversity of great minds that we have been having the privilege of hosting so far. So thank you all for your fantastic engagement in the series. Before I introduce today's speaker, I'd like to remind you that there will be a Q&A session directly after the talk, so please post your questions in the dedicated Slack channel and upvote questions you'd like to hear. We aim to keep the whole session to 1 hour in length. So it is my great pleasure to introduce today's speaker, Professor Judith Korb, Professor of Evolutionary Biology and Ecology at the University of Freiburg in Germany. Judith's work explores the evolution of sociality and the ecological and genomic mechanisms underlying cooperation, conflict, and reproductive division of labour in both natural and laboratory populations of termites. More recently, Judith leads an exciting and active research unit into the reversal of the fecundity-longevity trade-off in social insects, funded by the German Science Foundation, and she has revealed an intriguing role for transposon defense in determining termite longevity and aging. Judith has been elected to the Portal for Outstanding Female Scientists, Academia Net, a testament to her world-leading expertise in termite biology and her significant advancement of the field of social evolution. Today, Judith will present her talk entitled The Secret of a Long Fecund Life, Why Termite Queens Can Live So Long. So Judith, thank you for accepting our invitation, and over to you.
Okay. So thanks a lot for the invitation. It's really a great pleasure, and it's really great what you are doing, and to keep this going now since already a while. So it's really a lot of effort, and I think I enjoyed really very much to give this talk here, and it will be a great pleasure. And we have heard in the series already some talks about aging, and I think with my talk I will give it a social twist. And I hope by doing so and talking about social insects, I can show you that social insects have to offer some nice insights into aging and also into the evolution of life history. But before starting, I actually also want to introduce my two co-authors on this talk. This is Karin Meusemann, who is a postdoc in my lab, and you will see her picture showing up, and then you will see what she was doing for this talk. And then the other co-author is Daniel Elsner, who was a PhD student and who also did a lot of work on the talk that I'm presenting, and he's now also a postdoc in my lab. Okay, good. With this, I am going to start, and I will tell you something about the secrets of a long fecund life, and I will phrase it as a question at the beginning. So why can termite queens live so long? Okay, aging is actually really an evolutionary problem, because like an unlimited fecund lifespan would increase the fitness of individuals. So how can we explain it? Evolutionary theory explains aging by a declining force of selection with age. When we have a number of individuals and follow a cohort of individuals and follow them over time, there will be fewer and fewer individuals available or will survive over time. Most will die, so that we get at the end, at an old age, only a few individuals, and we get this selection shadow because selection will not be strong anymore. And because of this selection shadow, negative deleterious mutations that act late in life can accumulate over the generations. And this is the mutation accumulation theory to explain aging. Another theory related to this is the antagonistic pleiotropy theory. And this theory has in mind that genes that are pleiotrope, and such genes, when they have positive effects early in life and negative effects later in life, they will also not be selected against. So more or less what this theory all tells us is that mutations, negative mutations, can accumulate at old age, and when they are expressed, they are not selected against. A complementary theory to explain aging that also includes kind of proximate mechanisms is the disposable soma theory. And it sees aging mainly as an allocation trade-off. So individuals have a limited amount of resources, and they are selected to allocate them into growth, maintenance and repair, and reproduction. And if an individual has allocated resources for some of these functions, they will not be available for other functions. And as a consequence of this, life history trade-offs emerge, and the very common life history trade-off is the trade-off between fecundity and longevity. How much an individual should invest into which function and when should depend on factors like the extrinsic mortality. And as the soma is disposable, an investment into the soma, so meaning into maintenance of the body, should be limited in relation to the investment into the non-disposable soma. Some evidence in support for the disposable soma theory seems to come from studies on proximate mechanisms. They have revealed that the TOR, target of rapamycin, and the IIS, the insulin-like growth factor signaling pathways, are central for aging. These two pathways sense the availability of resources, and they regulate downstream processes, and therefore they regulate the allocation of resources into different processes. And by doing so, they interact with hormones like juvenile hormone JH in insects, or with other mechanisms linked to aging like oxidative stress. Okay. So I just mentioned that a major trade-off in animals is the fecundity longevity trade-off, when investment into fecundity is associated with a decline in longevity. And this trade-off is almost universal among animals. A major exception are the social insects, such as the social hymenopterans, ants, bees, and wasps, and the termites. Termites evolved eusociality independently of the social hymenoptera. And these social insects are characterized by a so-called reproductive division of labor. That means within a colony, only a few individuals reproduce, normally just one, the queen, and the other individuals forgo reproduction. And surprisingly now, it is the queens, so those individuals that are reproducing, which are the more long-lived individuals. So they seem to have overcome this fecundity longevity trade-off. And we will see now a very extreme example for this. This is the queen of a fungus-growing termite, so this individual here. And she actually can live for 20 years. This is a really very long lifespan for an insect. When you consider, for example, your pets, which maybe live around 10 years, or when you consider, for example, other insects, such as Drosophila, which just live for a few weeks. And during these 20 years, the queen also can produce 20,000 eggs per day. And this is an enormous number, and she can't produce more because this is the time it takes to press an egg out of the body. So these social insect queens are probably the organisms, the terrestrial organisms with the highest reproductive success. So they have a really high longevity and a really high fecundity. And on the other hand, within the same colony and sharing the same genetic background, because the queen is the mother of these individuals here, these are the workers, and these individuals just lived a few months and they are completely sterile. So within the research unit so long, we are actually studying this apparent reversal of the trade-off of the fecundity longevity trade-off in social insects. And we are doing this in a comparative approach, studying bees, ants, and termites. Termites, this is our part, and using Drosophila melanogaster as a solitary outgroup to compare with. And we are addressing in our research unit proximate mechanisms about aging and the reversal of the fecundity longevity trade-off, but also ultimate questions. Okay, so what I just told you, that there's a reversal of the trade-off in social insects, is actually not really true, because we haven't really shown this. What we have shown, or what I just told you about, is just a correlation between fecundity and longevity. In order to show that there's really no trade-off, we actually have to do manipulations. We have to increase fecundity and have to look whether this has an effect on longevity or not. And this we have actually to do for all the members within a colony, so meaning for the queens and also for the workers. Okay, and this is actually what we wanted to, or what we did in our first experiment that I wanted to show you. We wanted to increase the fecundity, and we used for this one of our model organisms, which is called Cryptothermus secundus. It occurs in mangroves in Australia, and it belongs to the so-called wood-dwelling termites. This means that it sits within such a dead piece of tree, and this piece of tree serves at the same time as food and as shelter. So these termites never leave their nest to forage outside; they just eat their whole house. And associated with this lifestyle is that they really have small colony. They are less socially complex. It's not like the other ones which I've just shown you before and which I will talk about later. And that the workers are really totipotent. This means the workers are actually totipotent immatures from which the reproductives develop. And this is important for the following of the talk. Okay, so what did we do with our experiments? So we wanted to increase fecundity. In order to do this, we removed eggs from some colonies. And we had control colonies, which we handled identically, but we didn't remove eggs. And this we did for a treatment period of 6 months. And after this treatment period, we had a post-treatment period for 12 months, where we just let the colony develop. And we did this design to have really a period where the queen fecundity was manipulated, and to test really whether egg removal increases fecundity. And we have this post-treatment period. And in the post-treatment period, we measured really what consequences the egg removal had. And we measured the survival of queens, the fecundity of queens after the treatment. And the fecundity of queens after the treatment is called the young offspring that were produced during this 12 months treatment. And then we also looked at the survival of the so-called old workers, and the old workers are those workers which were present during the treatment period. At the end of the 18 months, we actually killed the queen and one worker of 7 colonies from the egg removal experiment and 7 colonies from 7 control colonies. And we did this in order to generate transcriptomes to look at gene expression differences between treated and untreated colony. And this we did in order to see whether we see any aging effects associated with our treatment. So it might have been that the queens and the workers didn't die during the 12 months, but they actually were aging faster, and therefore we did this transcriptome experiment. In total, we had 37 experimental colonies for this setup. 16 colonies were egg removal and 21 were control colonies. And these experimental colonies derived from 8 stock colonies. Stock colonies means that these were colonies with different genetic background. And we did this design on purpose in order to test for treatment genetic background interactions, because we had then always from almost all colonies several replicates in the experimental colonies. Okay, so what are the results, or what were the results? So first, did the experiment work? So could we increase the fecundity of the queens by removing her eggs? And what we can see from this diagram is actually that it works. So what you see here on the y-axis is the production of eggs in control colonies and in egg removal colonies. And what you can see here is that queens where their eggs were removed laid around twice as many eggs as those in control colonies. So we could increase the fecundity of queens. Oops, sorry. So did this have the increased fecundity of the queens? Did this have an effect on the survival of the queens? And actually not. So you can see this here. So this is the survival of queens in control and in egg removal colonies. And in gray are the queens that died during the whole experiment. And you can see that there is no significant difference in the survival of queens, although some had an increased fecundity. What is about her post-treatment fecundity? So was her fecundity after the egg removal affected? And this we can see here, and we have to say, yes, it was affected. So queens which were treated, so where the eggs were removed, they produced fewer offspring than those in control colonies where the eggs weren't removed. But what we also could see in our experiment is actually that it depended on the genetic background. So from which stock colony the experimental colony derived, whether there was an effect or whether there was no effect. So you can see here, for example, these colonies from stock colony 1, they were hugely affected by egg removal, while other colonies here were not affected at all. So this means like that whether a queen was affected in her post-treatment fecundity depended on her genetic background. So we have now talked about this, fitness variables in the queens. So what about the workers? And we are looking here at old workers, so meaning those workers which were present during the experimental treatment. And we can also see here that the workers were affected. So those workers which were sitting in colonies where the queen had an increased fecundity were less likely to survive. And as like with the fecundity of the queen, we saw a strong effect of the stock colony. So meaning with some genetic backgrounds, this effect was observed, but not in other colonies which had another genetic background. Okay, I mentioned that we did, in addition to these direct fitness measures, we did transcriptomes and to look at differential gene expression to get insights whether queens were aging faster. We saw that the queens were not more likely to die. So did we find any evidence that the queens from the egg removal colonies, so those with an increased fecundity, had signs of aging? And therefore, we had seven queens from treated and from untreated colonies. And we looked at the so-called DHGs, so the differentially expressed genes. And actually, we were very surprised because we didn't find any difference in the expression between queens from egg removal and control colonies. So that means that we actually didn't find any evidence for aging of these, or faster aging of these queens in the egg removal colonies. So what about the workers from the same colonies? So actually the workers seem to be affected. So workers in the egg removal colonies had 110 genes which were significantly downregulated in egg removal compared to control, and 15 genes which were upregulated. So this means that actually the workers seem to be affected. And what are these genes doing? So many of these genes were related actually to development. And I mentioned at the beginning that workers are immature instars in this species, and that they can develop into reproductives. And so what we think that this signal might indicate that the worker's developmental trajectory is affected depending on how the queen was treated. So the workers themselves were not treated at all. So it's really just the queen effect. And therefore, what we also then wanted to know, how can this be that the workers are affected although the queen is treated? And therefore, we redid the experiment, or at least the first part of the experiment, with some other colonies. And this time we did behavioral observation. We observed workers and queens and kings within a colony and looked at their behavior. And we didn't find any difference in the behavior of the queens and the kings and the workers from control and egg removal colonies. The only effect that we saw was that workers fed queens longer via proctodeal prophylaxis in the egg removal colonies than in the controlled colonies. So what is this proctodeal prophylaxis? So this proctodeal prophylaxis is an anal feeding. And so that means that the queens were fed more or longer with anal secretions. So this might indicate that the workers kind of compensated some of the survival and also the reproductive success of the queen by feeding the queens longer. And this might explain why the workers were affected by the treatment of the queen. So when we sum up the first part of this experiment or of this talk, we can summarize that the fecundity manipulation was successful. So by removing eggs, queens produced more eggs. For the fitness of the queen seemed to be less affected. We didn't see any change. We didn't see any significant change in the survival of queens, and also the differential, the DEG, so the differentially expressed genes, didn't provide any evidence for increased aging because there just were no differentially expressed genes. The post-treatment fecundity of the queen were partially affected, and this depended on the genetic background. In some colonies, it was not affected at all. So for the workers, we saw that the workers' fitness was affected. Workers were less likely to survive. This also depended on the genetic background. And we also saw a signal in the transcriptome that genes linked to development were differentially expressed. So how can we And one thing is the mechanism that might be behind all this, why the workers are affected, might be because the workers feed the queens more via proctodeal prophylaxis in the egg removal experiment. And so how can we interpret all this? So this seems to indicate that workers take over partly the burden of queens. And this means that the trade that there is still a trade-off in this colony, but this trade-off is partially shifted from within an individual, the queens, to the workers. And this is in line with an idea about social insects functioning in superorganisms. I was very skeptical about them because there's a lot of criticism about superorganisms, which I still share. But I think it somehow supports it because, like, the queen functions as a kind of germline, and she is protected by the soma, with the workers which function as soma. So the workers take over partly the burden of the queens. With this, I'm coming to the second part of the talk, and this is more about the mechanisms of aging and the fecundity trade-off. And we are also using another model organism to study this. And this is the fungus growing Termite Macrothermus bellicosus. This comes from Africa, and this builds these huge mounds. And this is probably what you normally have in mind when you think about termites. And these termites are completely different to the ones which I talked just about. They really live in very complex societies with several million individuals. They also have the king and the queen, which are the one individuals that are reproducing. But this species also, they have major workers, which are males, and they are completely sterile, so they don't develop into reproductives anymore. And these major workers are mainly involved in foraging, so which means that their extrinsic mortality is really very high. On the other side, we have a separate caste. We also have minor workers. These minor workers are female. They are also sterile. They mainly stay inside the nest, so their mortality risk is lower. And these females, these minor worker females, can also develop into soldiers. We have in these colonies two castes of soldiers, majors and minors again, but I'm not going to talk about them anymore in the remaining of the talk. Okay, so I'm studying actually Macrothermus bellicosus since a very long time, actually since a student. And these studies were done in the Como National Park in Ivory Coast. And we know from studies by Rainy Leuthold that in the lab, such a queen can live for more than 20 years. During the field studies, we could actually also measure the survival of kings and queens in the field. And we did this using Kaplan-Meier survival analysis, and these data indicated that in the field in Africa, the median maximum longevity for Macrothermus bellicosus reproductives is 6 years after mound emergence. As these colonies live 3 to 4 years underground, the total median maximum longevity is 10 years for these kings and queens in the field. On the other hand, we have these workers, and these workers really just live a few months, 2 to 3 months, and major workers live shorter than minor workers because minor workers can develop into soldiers. So what did we do now in the study? So we again generated transcriptomes, and we were interested in mechanisms underlying aging. So we generated transcriptomes of different castes. So you see them here, major workers, minor workers, kings and queens, and compared these transcriptomes from old individuals indicated by this walking stick in the gray, and compared it to young individuals. And like for the reproductive, what is important to stress here is actually that the reproductives really were really old. So they were really 6 to 7 years old, or in total they were 10 years old. So meaning they were at the maximum longevity in the field. And so what were the results of this differential gene expression analysis? You can see this here. You see the number of differentially expressed genes. So in the first row, how many genes were upregulated in old individuals, and in the second row, and down, yeah, in both cases, always upregulated and in the young individuals. Okay, and what was really surprising when we got this result was actually that we didn't find many genes to be differentially expressed between the young and the old reproductives, between the young and old queens and the young and the old kings. So we didn't see much signs of aging in these reproductives. But we saw really, really many genes, over 5,000 genes, to be differentially expressed in young and old major workers, major workers. And you see this here. And then we had, of course, a detailed look in what are these genes doing that we found in the major workers to be differentially expressed. Everybody back again. I'm back again as well. So what I just mentioned was that we have what are these genes that are upregulated in these old major workers. And what we can see here is actually most of these genes, or many of these genes, more than 10% of these genes were transposable elements. So they are all listed here, and we see RNA as well as DNA transposons, and they are coming from different families, but marina transposons were especially prominent. Okay, what are these transposable elements, these TEs? So TEs are actually selfish elements. They have also been called jumping genes because they can translocate within the genome, so they can jump from one position in the genome to another position. And as you can imagine, is such a jump, or if they are active, these jumping genes, these TEs, they can really destroy proper gene regulation and gene function, and depending on where they insert. So these jumping genes, when active actually mess up everything in the genome. And because of this, they have also been associated with aging since a long time, and they have to be assumed to underlie aging in a range of organisms. And when we were doing the study, actually also experimental evidence from Drosophila melanogaster came out that actually jumping genes can cause aging, so they can be a cause of aging. And as you would imagine, if something is such deleterious for an organism, that mechanisms have evolved to silence these jumping genes. And one of these mechanisms are the so-called pi-RNA pathways. They are normally active, or the pi-RNA pathway is normally active in the germline of animals, and it becomes activated when transposons become active. So how does this pi-RNA defense work? And what I will show you now is a very simplified version, and especially the part of the ping-pong amplification cycle of pi-RNA defense. So if TEs are active, so-called pi-RNA clusters in the genomes get transcribed. And these pi-RNA clusters can be regarded as memories of past TE activity. And these transcripts of the pi-RNA clusters are then processed to produce antisense pi-RNA. And these antisense pi-RNA then binds active transposon RNA. And this duplex then leads to the cleavage of the TE and to the production of new sense pi-RNA, which is important for the transcription of pi-RNA cluster transcripts. So what we have here in essence is more or less a mechanism that destroys active TEs and generates more pi-RNA cluster transcripts to destroy even more active TEs. So we have a positive feedback mechanism to defend TEs. And important genes in this ping-pong amplification cycle are Argonaute-3, aubergine 1, and Quin. These genes are almost only associated with this ping-pong cycle. And we were looking now at these genes also in our transcriptomes, whether we found any signature here. And indeed, actually, we found a signature. So when we looked at the old major workers, aubergine 1 was downregulated in old major workers. And the same was the case for Quin. This implies that really the ping-pong cycle is downregulated in the old major workers. Further evidence for this comes from other genes, like zucchini, which was downregulated in old major workers, and capsulin, which was also downregulated in old major workers. But these genes also do other things besides being active in their ping-pong cycle. And what is now important is to know that these genes were only downregulated in the old major workers, but not in the old minor workers or in the old queens and kings. So this seemed to be something which is age-specific for the major workers. And how can we now interpret these results? And with this, I'm coming to the summary of the second part. So old major workers are associated with a high TE activity, and the high TE activity correlates with the downregulation of pi-RNA genes in the old major workers. These pi-RNA genes were not downregulated in old queens, kings, or minor workers, and hence we think that the constant upregulation of these pi-RNA genes in the other castes protect them against TE activity and probably against aging. And what was kind of surprising was that we actually did our transcriptomes from hats, and the pi-RNA pathway was described as a pathway that actually protects the germline. So this looked like a co-option of a germline defense system to be used in the soma of the kings, the queens, and the minor workers. But it has now been found that also in other organisms, it can be pi-RNA pathways can be up also in the head. Okay, and with this, I want to summarize both results. So what can we interpret from these results? Well, from the first experiment, the fecundity experiment, we have seen that workers partly take over the queen's burden, and so that the trade-off is actually shifted from within an individual, the queens of the fecundity longevity trade-off is shifted from within an individual to between individuals, from queens to workers. So we still have a trade-off. So it's not that there is a complete reversal of the fecundity longevity trade-off in social insects, but we have this trade-off more at a colony level than at the individual level. And in the second part, we could see that the germline defense mechanism protects the queens and the kings against TE activity, and TE activity is probably associated with aging in workers, in termites. With this, what can I summarize from this? Well, this provides again support for this kind of superorganism concept. So a colony actually functions as a superorganism. The queens are the germline, and therefore they are very well protected, as the germline is for an individual organisms. And the workers function more as the disposable soma, and therefore they are less well protected. And I would expect the degree how much this germline soma division is within a social insect colony depends on the degree of sociality. As long as workers can reproduce and become the germline of a colony, I think there should be, the shift should not be complete between workers and queens. Okay, so this provides some support for the disposal soma theory. So there seem to be allocation trade-offs between queens, kings, and workers. And yeah. And now to answer the question which I asked at the beginning, so why can termite queens and kings live so long? My current answer would be ultimately because they have workers that function as soma and partly take over burdens of the queens. And approximately because they are well protected against TE activity, but we also know already that other mechanisms probably play also a role in protecting the reproductive. With this, I'm at the end, and I want to thank you for listening, and sorry for the problems with the video. And I especially want to thank Daniel Elsner, who was a former PhD student and who did a lot of the work that I presented, and Karen Meusemann, who's a postdoc in my lab, and then also Volker Nehring and technicians in the lab that were heavily involved, and my group, and also all members of the research unit so long for many discussions and having a very fruitful time. Thank you a lot for your attention, and I'm happy to take questions.
Fantastic. Thank you very much, Judith. That was a really nice and interesting talk, and a lot of thought-provoking questions about how trade-offs can exist between individuals within a colony, and as you described, the germline being the reproductives and the soma being the workers. So it's really fascinating ideas. So we have a lot of questions from our audience, so I'll go straight on to asking you some of those. So we have a question about, do you think the queen-worker trade-off can be modified by changing the access to resources? And in your experiments, was there ad libitum access to food?
Yeah, so like in our experiment, there was ad libitum access to food. And we actually did an experiment where we limited also food availability. And this showed then also that queens and workers are affected. So then under food limited conditions, both castes are more affected also in their survival.
Ah, I see. That's really interesting. There's some interaction with the resource availability. So we have a question about the first part of your talk, and when you were talking about the termite species being totipotent, and the question asks, under what circumstances do the workers reproduce in our species?
Okay, so they normally never reproduce as long as a king or reproductive king or queen is present. But if the king or queen dies, a worker will develop into a replacement king or queen. Or if the colony sizes are very huge, which means in this species just 200, 300 individuals, the workers will leave the colony, so they will develop into winged sexuals and then colony. So these workers are really like just the young or the immatures of the king and the queen. And what this might be also interesting is like if a queen is not fecund anymore, she will be killed and will be replaced by one of her workers, which is then developing into a queen.
Oh, wow. It's exciting dynamics. So we have a question about the transcriptomics, and the question asks which tissues were used to assess transcriptomics, and especially in the first experiment where there are no signs of aging-related gene expression. Yeah.
So we used the head. So we used head transcriptomes in both experiments.
Okay.
So normally one would expect to see differences. Like we also see them when we do other experiments, we see them when we are using head. So like when we did, for example, this experiment, we see the expression from treated and untreated queens.
Oh, that's great. And related to the transposable elements, there's a question which asks if you could comment on the percentage of total gene expression that the transposable elements make up, not just the differentially expressed genes.
In total, how many genes in the genome are differentially are transposable elements, or is it like, because like it was of all differentially expressed genes, more than 10% were transposable elements, but this was only the case in the old major workers. So this was really something which was associated with the old major workers. So in the other in the other castes or in young individuals, the transposable elements are not expressed, or very few are expressed.
Yeah, that's great. So there's a question about could the future generation of queens be affected in terms of both longevity and fecundity? And how could this affect future colonies if the fecundity of the current queen is increased?
Okay, yeah, we don't know that, and it's really very difficult to do. So whether there are transgenerational effects, we just don't know that. But this is a real problem which we always have with almost all social insects, that the generation time is so long, and therefore it's really difficult to look at generation.
Yeah.
But it would be really interesting to do whether the future generation is affected.
Yeah. It's quite interesting how you saw an effect of genetic backgrounds. I was wondering what your thoughts were about why in some genetic backgrounds there was no trade-off found, whereas there was more of a significant effect in other backgrounds, and whether this could relate to particular elements of the genomic background or aspects which differ.
Yeah, so this is really also a very interesting question. We cannot answer it at the moment. We don't have any ideas. We just saw those colonies where the queens were affected in their fecundity, where the same colonies where also the workers were affected in their survival. So this correlated, but we don't know anything about the genetic background really, because a big problem is, like in social insect research, they are not model organisms. We have now a genome for the species, but we don't know very much. But this is something which I think is very common in our termites, because like we culture them exactly the same way, and nevertheless we see often high variances, high differences between colonies, even though they were treated the same way. So I think there is a lot of standing variation there on which evolution would act within the genome. And at the moment, as it's not a model organism, unfortunately, we can't tell what the causes are, but it would be really interesting to know.
Yeah, definitely. That's quite exciting. So there's lots of people who are quite interested in the superorganism idea, and the fact that the trade-offs seem to exist after all between longevity and fecundity, but it was between different individuals within the colony, so at the colony level. I was just wondering if you could talk a little bit more about criticisms of the superorganism idea, or I think you mentioned that briefly. I was quite curious.
I think like the big problem with the superorganism is that that the individuals within a colony are not to 100% related. So they are not clones. This is different compared to an organism. And therefore we have also conflict within social insect colonies, which we normally don't have within cells within an individual. And I think this is, from my point of view, this is the biggest criticism on the superorganism concept. And I still see this like this. So the social insect colonies are not really functioning like organisms. So there are conflicts. And in other work, we have shown that there are conflicts. But like in this regard, they somehow function as a superorganism. And I would predict, this is something which we are looking at the moment into it, that it really is dependent on whether workers can reproduce or not, how superorganism-like these social insect colonies are. And I would predict, for example, like when the workers are completely sterile, the whole trade-off can shift it from, or the whole burden from the queen can shift it to the workers. And then we, of course, like when the workers are sterile, there is no evolutionary interest in becoming a reproductive, and therefore I think they would, under such circumstances, they would function really as a disposable soma. Whereas in the first species which we looked at, where the workers can reproduce, they are not really like a completely disposable soma, because they can develop into reproductives. And there are also more conflicts in the first species than in the second.
Ah, I see. That's really interesting. So we're just finished with one quick final question for the moment. So, and there's a question about how you measured survival for the Cryptotermes experiments. Yeah, basically.
Yeah, so we can trace, so the queens, they dig into the wood, so we can't see them, but we can see whether a queen survived or not by their sclerotization. If a queen died, then a new replacement queen will develop from the workers, but it's much lighter sclerotized. And this was also the reason for the setup of the experiments, that we did the second part only for 12 months, because after 12 months we are still sure that we can differentiate between old queens and young queens by the sclerotization. So this was the reason for the 12 months post-treatment period.
Ah, I see. That's a clever trick. Great. So I think that's all we have time for today, but it is our great pleasure to thank Judith once again for a truly fascinating talk. Thank you very much.
Thank you. Thank you for hosting me. And yeah, I'm looking forward to more talks. Thanks a lot.
Thank you. So our next talk will be from Professor Rosemary Grant on speciation and Darwin's questions. So we hope that you can join us on Wednesday at the same time and same place. And in the meantime, check out our updates on Slack and our Twitter feed and spread the word about our seminars. Thanks for taking part. And until next time, stay safe and see you again soon. Goodbye.